is Dr. Gavin Lear from the University of Auckland, and Gavin's going to be talking about, as we said, organisation micro procedures for the education of life from environmental DNA.
Okay, long title.
That is a long title.
So I'm not sure who decided to call this session Disruptive Technologies. I feel like I need to apologise in advance in case I'm not disruptive enough, but there we go. So what I'm doing in this session is I'm going to outline my efforts and the efforts of this motley crew as we try to recommend and standardise molecular approaches for the identification of wildlife from environmental DNA. So we've had a little introduction already to what environmental DNA is, but as I'm the first speaker in this session really focusing on this topic, I've got the luxury of providing a little bit more detail here. So environmental DNA is the DNA, ooh, we've got some problems here. There shouldn't be any timings on this, we might struggle. Environmental DNA is DNA that's collected from an environmental sample. So DNA that's shared in skin, hair, and so on. So the best way to kind of look at how important environmental DNA is probably to consider this in a human context to start off with. So you're looking like a fairly clean bunch. Regardless, as you sit through this session, each of you is going to shed probably tens of thousands of skin cells, so much that maybe every month you're essentially going to rejuvenate your entire skin. You're going to get through about half a kilo of skin cells a year, and if you get through your entire life and live to about 80, which we will hopefully hope to do, you'll shed about 1,000 skins, if you want to put it in that way. So as we go through our life, we shed a lot of material into the environment from our skin. What happens inside us is even more dramatic. So, for example, here we've got the epithelial cells within the gut. They're generated and cast off at an amazing rate. Every day you're getting through about 100 billion. Even a little mouse gets through about 100 million of these cells. So hopefully I'm not talking about the audience now, but as, for example, an animal goes around the environment, shedding its skin, weeing and pooing everywhere, we can trace where these organisms are by looking at where that DNA ends up. It's no different for plants. They shed enormous amounts of DNA through their leaf litter and through their root exudates as well. So why do we actually want to use DNA that's cast into the environment to monitor biodiversity? Why can't we just use traditional techniques? Often a reason for this is because lots of species and subspecies actually look quite similar. I'm a microbiologist. I can't tell you if this is a Hector's or a Maoist dolphin. If you give me the DNA, I might have a bit of a better shot. Other reasons is because we don't actually need the organisms to be present in the environment for us to know that they've been there. For example, eDNA could be used quite easily to track the distribution, for example, of nocturnal species, such as this Asian fishing cat here, possums maybe, or to track the distribution of organisms that hide from us when we try and sample. So we don't need the organisms to be present in the environment to know it's passed through there. Another thing with eDNA is it's fairly non-destructive to sample. If you think about how we sample biodiversity at the moment, for example, freshwater samples, to look at what insects are present, we have to go through that environment, kick the crap out of the stream, see what insects we can find. If we're looking at fish, we might electrocute that stream water to see what fish we can find. Doesn't sound very good when we might be looking to see what rare fish we have present in those environments. If we think of eDNA instead, all we need to do is take a small amount of water, see what organisms are present from the DNA that's there. So there's a number of advantages of using eDNA to monitor biodiversity. I use eDNA to monitor biodiversity because it's quick and easy. So, for example, I have groups of people who are not particularly experts in eDNA, collecting thousands of samples, soil samples, freshwater samples from around the country for me. They can collect these samples with ease and then send them to me in the post. So sampling is easy and relatively non-destructive. But the key advantage of working with eDNA is you can simultaneously study the occurrence of any kind of organism you want from a single sample. So, for example, if we've got a soil sample, we can extract all of the DNA from within that sample. Once we've got all of the DNA extracted, we can then decide, okay, what I want to focus on today is to work out what bacteria are present in that sample. So from that big pool of DNA we've extracted from that sample, we can then amplify a particular gene. For the bacteria, it would be the 16S RNA gene, and that will tell us what bacteria are present in that sample. Equally, I could give that sample to someone else, and they could look at what plants were present in that locality. Okay, so from single samples, we can look at the diversity across multiple domains of life. This all sounds pretty easy so far, hopefully. Why haven't we done it so far? One of the key reasons is, of course, that DNA sequencing has become much cheaper. So we've had an explosion in our abilities to sequence DNA and a corresponding decrease in the cost of our DNA sampling. That's one reason. But this is the big one here for eDNA, is that the advances in the technology mean we can now multiplex lots of samples. That is, we can simultaneously analyze the DNA from many hundreds, possibly even thousands, that's maybe a slight exaggeration, of samples in a single run. So actually looking at eDNA now is cost-effective, and when I undertake big studies looking at hundreds or thousands of samples, it's monitoring the pH, monitoring the carbon that might be present in that solar nitrogen that actually scares me, the cost of that, compared to the DNA analysis, which is quite cheap in comparison. Okay, does this stuff work? I'm going to cherry pick some good results here because that's always an easy thing to do. Here's a nice study that was undertaken in the Okinawa aquarium in Japan. By taking a few litres of water from a number of different tanks, and these eDNA scientists could identify 93% of the species present in those tanks within quite a short amount of time. Now, if any of you are feeling a bit miserable and pessimistic this morning, you might think about what about the other 7%? Well, actually, I think if you had a fish expert wandering around these tanks, they probably would struggle to identify all of those fish in a short amount of time. Yeah, this is hopefully, for many cases, quite an effective technique to monitor biodiversity. Okay, an example from the terrestrial environment. This is a New Zealand example. This was a study conducted on Little Barrier Island, and this was comparing biological diversity by, for example, looking at the trees and the seedlings present on the island with the data collected from eDNA, without explaining what these figures mean. The spatial variation in the vegetation was similar, comparing sample data collected by observation as compared to the eDNA data. So there was a correlation between those methods. Same for the eDNA data, comparisons with invertebrates, which were studied by eye. Thank you. Okay, so eDNA, also, it's nice to use eDNA to track the biodiversity of a landscape, but actually it's also quite interesting for us to use this to target particular species of interest. So within New Zealand, we don't have Burmese python present yet, but you're probably aware that within the Everglades in Florida, these species are particularly problematic. Even though there's loads of them, we don't quite have a good understanding of their distribution because they're well camouflaged and they spend a lot of time underwater. Sampling with eDNA, however, scientists have reported detection probabilities of 90% plus for Burmese python. That is, with a litre of water from that environment, you've got 90% plus certainty that you've got a Burmese python in the vicinity. Okay, so with this in mind, what we've been trying to do is to develop a national framework for biological heritage assessment using eDNA, and this is working as part of a project led by Robbie Holdaway. So that is to use existing biodiversity monitoring strategies, for example, biodiversity strategies undertaken by DOC, regional councils, and so on, and to expand on them by analysing and comparing DNA data collected from these different sites. So the idea is we can start to share the way that data and samples are collected to build up a picture of biodiversity in New Zealand from this eDNA data. Okay, again, hopefully this sounds fairly simple. This is starting to get a little bit annoying. The problems that we have found is that although we want to compare all of these sample data collected from around New Zealand, what we know is that everyone in New Zealand, for example, extracts the DNA from these environments in different ways, which is really annoying. There's so many different techniques out there to extract DNA. Everyone is using different techniques to do this. Similarly, even when we're looking at particular organisms of interest, for example, we might look at our fish here, different researchers will target different gene regions to study the diversity of fish looking at these eDNA samples. So if we have different researchers in different locations extracting the DNA in different ways and targeting different genes, it makes it really hard for us to mesh all this together to get a good picture of New Zealand's biodiversity because there's bias in the way that these different approaches are used. So we have tried to come around this to identify and devise a standard approach for eDNA monitoring within New Zealand. So we looked at all of the research that's been conducted to date to try and work out what techniques are in common use. And we held a workshop back in February 2016 to try and standardise our DNA approaches for extraction and amplification around New Zealand. So based on what people are using at the moment and for the knowledge in the room, we managed to identify, for example, one particular kit, this mobile soil kit, as being really good for extracting DNA from the largest range of different environmental samples. And also this sister kit here was very good at extracting DNA from soil. So they're widely used, and they appear to be the best for dealing with difficult samples. So for almost all samples that you deal with, including plant material, it would seem that this approach is quite appropriate. The only deviation from this was when we're extracting DNA from blood and tissue. We then went through, looked at all the different genes and gene regions that people were focusing on in their eDNA studies, and for some of these we were able to make recommends very easily. For example, when we're looking at our bacterial communities, most people were focusing on 16S RNA gene. For those of you that are interested, for the bacteria we decided 16S is best. For fungi, ITS2. For plants, another ITS region. For animals, the CO1 gene. And for fish, the 12S RNA gene. So we looked at what people were using so far to try and build in consistency, and we did some basic informatics, bioinformatic analysis to work out which genes were most appropriate. For example, here, the bacterial one, the one focusing on bacteria and archaea, works well. Identifies lots of them, but not so many of the eukaryotes to the right there. So, getting to the end, we managed to develop full protocols for the amplification of DNA from a range of different taxa. So for fish, bacteria, animals, and so on, we've developed these full protocols that will be published soon, so that you as users can follow these protocols if you want to, so that we can all develop a standardized approach to our eDNA sampling and DNA processing around New Zealand. You don't have to follow these techniques, but they're there for you to use. And I think a lot of people will appreciate these techniques being available because there are so many options to us as eDNA researchers, particularly for newer researchers coming on board. It's really nice to have each of these individual protocols available for people to follow, again, so that we can become part of a larger group of scientists all working together and creating data so that we're comparing, if you like, apples with apples. I'm going over time, so I'm going to whiz over this and just identify my future challenge. In lots of ways, this stuff isn't that challenging. The technology, you'd say, already exists. The biggest challenge is actually getting us to work together, looking and processing samples in the same way. However, one key area that we need to focus on is at the moment there's no centralized facility for eDNA storage. And I think that that is a big problem within New Zealand. What we need is a single centralized facility to archive our DNA. This will allow us to share sample material that's been collected among researchers. It allows us to preserve our DNA for resampling. And actually, I think this is one of the biggest things here. It allows us direct comparison with samples not yet collected. So think about some of the big advances, in science. I'm thinking about things like HIV. How do we know where HIV developed, how it spread across the world? It's because we had samples stored in freezers that were then able to analyze even many years after these samples were stored. They were stored before we even knew what HIV was. Think about forensics. We've managed to put criminals away by looking at DNA that was stored in freezers before we even knew how to analyze DNA. It's really important that we can have these stores of DNA as an asset for the future, so that in the future when people ask us what might be simple questions, such as when did Cody dieback arrive in New Zealand, we can say something that's a little bit more intuitive than I'm not quite sure. This will allow us to go back in time and work out when different diseases, for example, arrived on our shores. Okay, it's really hard to work with all of these different scientists working on these different organisms. One of the big challenges was us all putting aside our egos, not working with the techniques that we're necessarily always used to using, coming together as a big community to do this. So I just want to thank all of the guys that helped us to produce this standardized approach. Apologize for my slides doing crazy things. I guess that's part of the Crazy and Ambitious conference. Thank you.
Thanks very much, Gavin. We've got time for a question, I think.
What's the normal fate of DNA in the environment? How long does it last? And why hasn't it built up and taken over?
So people often ask this question, how long does DNA last in the environment? It depends how well it's preserved and it's stored, so it's not an answer that can be easily addressed in surface water. It might last a few days before it gets sheared. But DNA, for example, can bind to humic material, bind to sediment, and in that environment it lasts a lot longer. So it depends on what state that DNA is in. We know that in the environment DNA can last a long time. If you think, for example, we can get DNA sequence from marbones. So it just depends where it is. So it's really hard question to answer, but it's something that we do need to keep working towards to understand. Yeah.
Okay, one more. Right at the back, please.
Yeah, I'm from the biosecurity community, and there's one thing that I'm a bit confused about with the eDNA is its ability to get down to taxonomic levels. So, is it species, is it genus, or is it
Okay, so the question is, what kind of level of identity are we working at, species, genus level, and so on with our eDNA data? So we can kind of do what we want. What I think of when we're doing these DNA surveys is it's really good for us to get a broad idea of the diversity in our sample material. But if, for example, we find a new incursion of an organism, and we can identify that sample as being of particular interest, it will certainly be then of interest for us to go and analyse that sample more in depth, perhaps using different primers to species level within that sample to get more certainty as to how well that organism has been identified, and actually to go to that location to check that you can find the organism as well. So in short, what I would suggest is eDNA is really good as a screening approach, but to definitely say you've got an incursion of an organism from a single sample, I wouldn't trust it for that. I don't know if that goes part way to addressing your question. Yeah. Okay.
All right, thanks very much again.
Thank you.